Welcome back, class. Mr. Rasmussen here. This is lesson 9B. And in 9B, as you can see right above my head, right up there, you can see that we're going to talk about genetics and genetic crosses. So we're going to learn how to do genetic crosses in this. There's a lot of vocab that we have to have in order to make this work. So there's a lot of vocab in this chapter that we have to get a good understanding of what it means in order to become proficient at dealing with genetic cross problems. Now, before I start talking about genetics and genetic crosses, I kind of want to start by proposing a different problem. Say, for instance, I have this jar of gumballs here. And I ask you to tell me, if I put a quarter in there, what is the chance that I would get a pink gumball? Well, in order to solve this problem, you may count up the number of gumballs and figure out the number of gumballs in there that are pink. And therefore, you can figure out what percentage of gumballs are pink, and therefore, you would know what percentage of a chance I would have to get it. But that doesn't mean, say the chance is 10%. That doesn't mean, though, that if I get 10 gumballs, I'm necessarily going to get a pink one, even though the chance is 10%, because I only am working with a probability of getting a gumball every 10%. That doesn't necessarily mean it's going to happen. The same thing with flipping a coin. If I take a quarter here and I flip that quarter, if I flip that quarter once, I know that I have a 50-50 chance of landing on either heads or tails. But if I flip that twice, that doesn't mean that I'm going to get one heads and one tails. I may flip it twice tails both times. So that's probability. So in genetics, we're going to be dealing with probability. We'll never know the exact outcome of a cross. Most of the time. Sometimes we can tell, and I'll show you that. But most of the time, we're never going to know the exact outcome. It's only a probability. Well, they're going to have a chance of having a child that looks like this. So we're going to be dealing with probability and chance a lot in genetic crosses. I kind of want to start with a familiar picture to us. We saw this one here, and I want to talk a little bit about alleles and genes. So if we notice here, we have two homologous pairs of chromosomes. Right? It says here homologous pairs of chromosomes. And this is chromosome number 1, and this is chromosome number 2. So we have a pair of homologous chromosomes, and they're the same. And they contain the flower color gene, the gene for flower color, on both chromosomes. So on this one here, it contains the genes for flower color, and over here, right here, it also contains the genes for flower color. So both of these chromosomes contain the gene for flower color. So there are two pieces of information for each flower color gene. Two pieces, one on each homologous chromosome. And we call that an allele. An allele is just a piece of information for a gene, and there are two alleles. Two alleles, and they're found on each of the two chromosomes. Okay? This allele over here, if we look at this, white flowers in pea plants ends up being recessive. So we would give it if we were writing this, our shorthand for it, we would say that that allele is a lowercase. Now it doesn't matter what letter we use. It doesn't matter what letter, if I use an A or a V or a T, it doesn't really matter. What matters is the case is correct. So white flowers, which is recessive, gets the lowercase allele. This gene over here, which is dominant, purple flowers is dominant, it gets the uppercase allele, or the capital letter. So this is what the genes for this plant look like for flower color. They have one lowercase allele and one uppercase allele. So that's what their genes look like. That's where they're found. When we do meiosis, we know that you're only going to give one chromosome of each homologous pair to your offspring. So therefore, Mendel's right. You're only ever going to give half your genes to your children, because you're only ever going to give them one of each homologous pair of chromosomes. So that's where genes are located. A gene is just a location on a chromosome. The alleles are the different pieces of information for that gene. There are two alleles for each gene, and they're each found on a homologous chromosome. Now these alleles we give names to. Some more vocab for us. We have these two terms that you're going to see a lot come up, genotype and phenotype. Genotype, the term genotype, if we look back here at this here, we'll get to genotype in just a bit. If I look back here at this thing that we just did, so this flower here, that is purple and white, or has one violet gene, one dominant and one recessive gene, if we're looking back at that flower. The genotype for this flower, the genotype is what genes the flower has. The actual letters for the alleles. So the genotype for this flower would be, this is actually wrong. I'm sorry. It is not big A, little a. The genotype is actually, it's not big A, big A. The genotype is actually big A, little a. So we had one dominant and one recessive gene. And in our genotypes, we always write the dominant first. It's just a shorthand that we always do. The other term is phenotype. And the phenotype is what we physically see with our eyes. And in this case, because that dominant gene exists, because that violet gene, which is dominant, exists, we are going to see with our own eyes a violet flower. So the genotype for this flower would be big A, little a. It would be a term we call heterozygous, and we'll talk about that. But its phenotype would be violet flowers. Okay? Genotype is what genes, phenotype is what we physically see with our eyes. If we move on from there, let's talk about these terms homozygous and heterozygous. Now I'm talking about only genotypes here, only genotypes. Our genotypes can either be homozygous or heterozygous. What do I mean by that? A homozygous genotype has two of the same alleles. So for instance, a homozygous dominant genotype would be this one here. Big A is homozygous dominant because we have two of the same dominant alleles. Homozygous recessive looks like this, big A, little a. Two of the same alleles, but in this case they're the recessive alleles. Heterozygous is our other term. These prefixes here, homo, you'll see that show up a lot in biology. The prefix homo means same, and the prefix hetero means different. Heterozygous then means two different alleles. This would be the only heterozygous case for this gene. The only heterozygous case would be one capital and one lowercase, and just note that we always write the capital letters the same. You're going to want to sometimes write two different alleles. So if we're looking at flower color, you might want to say, oh, they have one violet, which is dominant, and one white. So you're going to write VW. That's wrong. We always keep the letters the same and just use the capitalization to determine whether it's dominant or recessive. So that's homozygous versus heterozygous. Does that make sense? Okay, moving on from there. I want to look at genetics crosses. I want to give you some tools to solve genetics crosses so that when we come into class tomorrow, we can work on solving some of these genetics problems and we can become experts at them and be able to conquer anything that we see. So, the way we solve genetics crosses is we use Punnett squares. Punnett squares just shows us the probabilities of having different outcomes of genetic crosses. So up here, outside of the Punnett square, right here and right here, are the genotypes of the parents. So this parent is heterozygous. They have one big A and one little a. This parent over here is homozygous recessive. They have two little a's. When we do the genetics cross, what we do is we take this where it lines up, where this column meets this row, so right here and then right here. So we take and put those two letters together. So we notice that one probability or one offspring possibility could be that that offspring shows up to be heterozygous. Another one down here, if we do the same cross, we take this A and we carry it down here in this little a, we notice that this child is also going to be heterozygous. So if we look over here, we cross this A over here with this A, so this A comes down and this A comes all the way over. We notice that we have two little a's, so that this child is going to be homozygous recessive. And if we look over here, this child is also going to be homozygous recessive. So the chance of us having a homozygous recessive offspring is 50%. Half of our children, we can see that by the green blocks here, half of our children are going to be homozygous recessive for the trait. And then over here we have half of our children are going to be heterozygous for the trait. So we don't know what we're going to have. If we have a child, that doesn't guarantee us that we are going to be homozygous recessive, but we know that it's going to be 50%. We're going to get 50% of our offspring being homozygous recessive. Okay? Let's look at one of these simple Punnett squares, and we call these a monohybrid cross. In a monohybrid cross, we are looking at only one trait at a time. So we're going to look at only flower color, or only height of a plant, or only one trait at a time. So here's our problem. It says a heterozygous violet flower is crossed with a homozygous recessive white flower. What is the chance that the offspring will be a violet flower? Where I want you to start here is the parents. We need to figure out the parents first. And I'm giving us some weird terms to describe them. A heterozygous violet flower. So we need to figure out what that parent looks like for the flower colored gene. Well, I tell you that that parent is heterozygous. So we know that one parent is big A, little a. And I'll use A's here. Okay, and we're crossing that, so it's crossed. So we're crossing that with another plant. And that plant is homozygous recessive for the flower color gene. Well, I know that there's only one homozygous recessive, so I know that it has to have two little a's. If where I'm getting these are confusing, flip back a couple, re-watch the things on homozygous and heterozygous, just re-get used to those terminology, because all I'm doing is using those. Homozygous recessive has to look like this, heterozygous has to look like that. So that's the first place to start. One, I would start by figuring out the parents. Two, you're going to draw a Punnett square. So down here, your Punnett square is going to look like this. Okay? I'm doing this with the mouse here, so if it shows up a little goofy, I'm getting used to that. So over here, I'm going to take one parent, and I'm going to align them outside of the Punnett square. So I'm going to take this parent, the first parent, and I'm going to put it up here. So I'm going to write this big A here, and this little a here. Okay, because I know that parent can donate either a big A or a little a to its offspring. Then I'm going to take this parent, and I'm going to put this parent down here, and I know that this parent can either donate that first little a or the second little a. Okay, and then when I cross these, we take this big A and match it up with this little a here. Okay, and I notice that if I do the rows and columns, this A meets up with this A, so I have this A here. I notice that this big A meets up with this A here, so I have big A, little a, and I notice that this A meets up with this A here, so I have two little a's. Now, that means, now I have my Punnett square finished, I can answer the question. It says, what is the chance that the offspring will be a violet flower? Well, if I look at this, if violet is dominant, this flower here, because it has that dominant allele, is going to appear violet. This flower here, because it has the dominant allele, is going to appear violet. This flower here lacks the dominant allele. It's not going to look violet at all. This flower is not going to be violet. So if I put just a dot in these two are going to appear violet. So I know that 2 out of 4 are going to be violet, which means that 50% is the chance. You'll have a 50% chance of these two flowers breeding, of producing another flower that has another plant that has violet flowers. Okay? That's a monohybrid cross. So we're only looking at one trait. That's a great place to start. We'll deal with monohybrid cross problems in class, but that's where we'll start.